So the first sort of theme that weave this way through is that of above-ground, below-ground linkages. The recognition that the things we see, the plants and animals that we see above ground, influence the organisms that we see in the soil, both in terms of those organisms that carry out decomposition and nutrient cycling, as well as those organisms more tightly associated with plant roots. And these organisms interact to see what we see above ground, with these above ground and below ground linkages driving functions above ground like production, and below ground like nutrient cycling and the second theme that pops up is that of generally state factors of soil formation. Plants grow in soil, we're therefore very interested in soil. And anybody that's done an undergraduate course in soil science will have very early on have been subjected to this particular framework. The recognition that soils are formed by 5 factors that collectively drive the formation of the soil. Organisms, topography, climate, the parent material, the rocks that form the soil in the first place, and time since formation. And these 5 factors will be making guest appearances at various stages throughout my talk. And the third theme is that of timescales. Now, the x-axis here is time. The processes that my colleagues and myself look at are short-term processes, plant-soil, plant-animal interactions, plant-plant interactions, things we look at with experiments. Things over the order of hours to weeks to years. But these interact with much longer term processes beyond our lifespan. Things like vegetation change, soil processes, geological process, and macroevolution. So the processes that we look at both respond and drive these longer term processes. I see there's a couple of people in the waiting room that somebody probably wants to let in. I'm getting these notifications popping up.
Yeah, sorry, we're doing it.
That's okay. So in my presentation, I'm going to go through 3 pieces of work we've been carrying out. First of all, work we've been carrying out over the past quarter century on lake islands in northern Sweden. Secondly, I'm going to say something about soil aging and chronosequences. Chronosequences are simply sites of different age since their formation, soils of different ages. So, and then the third, I'll talk about some of our work on climatic gradients in mountains. So starting off with the island system, the work I'm going to describe now is work in boreal forests in Swedish lake islands. And it's in the boreal zone. And in the boreal zone, by way of context, the main natural disturbance regime is fire. So boreal forests burn typically every 50 to 100 years if people don't get involved. Some parts of the boreal, the fire frequency is increasing through global climate change and warming. Other parts it's decreasing because people live in them and therefore they deliberately suppress the fires. These fires occur across, mostly across the ground and consume carbon and release carbon. So they determine the extent to which the boreal forest serves as a sink versus a source of carbon. And this is relevant because boreal forests actually store more carbon than either the tropical or temperate forests. And most of this carbon is stored in the soil. So the change in fire regime through human activity has a direct implication for the ability of boreal forests to serve as a sink versus source of carbon. Our study system is a group of islands in northern Sweden. They're formed by the retreat of land ice about 9,000 years ago. And they're subjected to the same extrinsic factors other than fire. So big islands are a large target. They get hit often by lightning, and some have burned in the last 50 years. So they're dominated by early successional plants, plants which grow fast, like Scots pine, Pinus, and Vaccinium or bilberry. Small islands are a much littler target. They get hit less often by lightning. Some have not burned for 5,000 years. So they have different vegetation that's adapted for nutrient poor conditions, much slower vegetation like Picea or spruce and Impetrum or crowberry. So what we essentially have is a sequence of sites. We've got big islands, frequent fire, fast-growing plants such as pine and bilberry, middle-sized islands, birch and lingonberry, and small islands, slow-growing plants which produce well-defended leaves and plant litter, grow slowly, spruce and crowberry. So this represents some of the very earliest work we carried out on the system. We sought to essentially study how island size affected various ecosystem processes. So this shows litter decomposition, which essentially drives carbon storage. is the decomposition of dead leaves placed on the soil surface and buried in the soil. And what we find is that big islands with fast-growing litter, plants that produce fast-growing and produce fast decomposing litter, have dead leaves that microbes like to break down, compared with on the smaller islands where processes are considerably slower. And this in turn influences carbon storage on the islands. What this shows is carbon storage versus island size. For the large islands with rapid decomposition, we have really shallow humus, organic matter of just a few centimeters in depth, whereas on the large islands, decomposition is slowed down and we end up with really deep humus profiles of up to 1 meter or more. And this is also apparent if you plot out carbon storage in terms of time since last fire using carbon-14 dating of our charcoal from each island. Over the 5,000 years since fire, carbon accumulates at a more or less linear and unabated rate and doesn't reach any sort of asymptote. Now, this is relevant to global carbon issues. It has been proposed that suppression of fires in northern forests could be sequestering carbon and explain some of the so-called missing carbon. Carbon that's released by the burning of fossil fuels that remains unaccounted for in global carbon budgets. If we get results like ours and extrapolate them over the entire northern forested zone, you can show that even 200 or 300 years without fire causes enough carbon storage to explain some of the so-called missing carbon. Now, this is as far as the story got 10 years ago. This is work headed up by my then postdoc, Mikkel Jonsson, who actually gave a seminar at the ASC a couple of years ago. But what this shows is a structural equation model. Basically, the thickness of an arrow indicates the importance of a pathway. So on our islands, time since fire has strong direct effects. Fire burns carbon, less fire, there's more carbon. There's an effect through vegetation. The types of plants we have and their attributes affect carbon, like I mentioned in the last few slides. And there's also a relatively weak effect through plant diversity. Plant diversity is generally recognised in promoting carbon storage. Now, we thought this was the complete story up until 10 years ago, but then we started working more with people who looked more at root systems, and we then got to learn that this system is actually very incomplete and kind of inaccurate. The problem emerges through work carried out by then postdoc Carina Clementson, who led a modelling exercise of our island system. What we found, what this shows, this is the result of the modelling exercise, is if you follow this curve for the large islands, this is the amount of carbon that down the soil profile that can be explained by plant litter, production, decomposition, litter quality, from what we know of litter production and litter decay dynamics from the system. This dashed line is carbon down the soil profile that cannot be explained by plant litter and mustn't come from somewhere else, such as pumped underneath from plant roots. So after about 20 years of age down the profile, most carbon is root derived. And this is even more apparent on the small islands with the really deep humus. This is carbon down the profile explained by litter. This is carbon that could not have come from litter and came from roots. Most of the carbon is actually root derived. And we have multiple lines of evidence that this carbon is actually not derived from the plant root material from the plant itself, but actually from fungi that are associated with the roots. And we have multiple lines of evidence for this. This diagram is not as complex as it looks, and I'll walk you slowly through it. The y-axis here is soil depth as we get to the older soil. Solid line is the big islands with the shallow humus. Dotted line is the small islands with the deep humus. R is what we know for plant roots. M is what we know for fungi, or specifically mycorrhizal fungi, special fungi that link with plant roots. So as we go down the soil profile, the stable isotope of the carbon gets closer to that of fungi and not roots. Same with regard to nitrogen, closer to that of fungal material and not root material. And the same also with the carbon to nitrogen ratio down the profile, closer to that of fungi, particularly for the small islands with the really deep humus. We get further insights from ergosterol, which is something that fungi produce. This is free ergosterol, so it breaks down really easily. Lots of it on the large islands with the shallow humus, not so much on the small islands with the deep humus. However, if we look at bound ergosterol, ergosterol is tied up in tough forms. Lots on the small islands with deep humus, not so much on the large islands. So the deep humus is because we've got lots of this tough fungal material and not so much of this very easily breakable, downable fungal material. And finally, if we look at glucosamine, which is the active protein constituent of fungi, lots of the subsoil of the small islands. Now, we've taken this further to look at the types of fungi that lead to the organic matter accumulation. This shows an ordination diagram of all of our soil samples in terms of their fungal communities. For those not familiar with ordinations, basically points that are closer together have more similar fungal communities, further apart they're more different. So big islands are big circles are big islands, little circles. Blue is soil deeper down the soil profile, green is nearer the soil surface. So we find that big islands have fungi that are associated with high production, low soil carbon. The little islands, low production, high carbon. This here is the same diagram that I put in my previous slide. And these other three diagrams are subsets of the soil fungal community plotted out in exactly the same ordination space. So this here are special fungi called basidiomycetes. They're associated with trees and they produce dead material that breaks down really fast. Lots of them on the big islands with a shallow humus. These are other fungi we call ascomycetes. They produce residues that are really tough, well defended, break down really slowly. Lots on the small islands with a deep humus, not so much the big islands. So the deep humus in the small islands is due to lots of these guys and not so many of these. And what this shows is fungi that are associated with dead leaves, what we call saprotrophs. Lots of the upper part of the soil profile, which if you recall from our model, is the only part of the soil profile where we would expect organic matter to be formed by dead leaves as opposed to dead fungal material. So with this more complicated fungal-driven version, this is now how we understand carbon to be stored in our system. If we look here, this is the large islands with basidiomycetes that produce residues that break down fast. They release lots of CO2, lots of nitrogen, so the plants are happy because they have lots of nutrients. Shallow organic matter. Small islands, the fungi produce resistant structures that break down really slowly. Not much carbon and nitrogen given off, so the plants look a bit miserable, but lots of carbon and nitrogen stored in the soil. Deep humus profiles. This is relevant to global change. The more frequent fires we get in the warmer parts, in the parts of the boreal with more frequent fires, move to this direction. Suppression of fire and least frequent fires, we move more in this direction. Now we've also been looking at how the different components of biodiversity in the system drive carbon storage. And what I'll describe just briefly is an experiment we've been carrying out now for the past 24 years on each of 30 islands, looking at where we manipulate vegetation. So on each island, we have two sub-experiments. The first of these focuses on three plant functional groups. Trees, which we manipulate below ground by root trenching, we cut their roots off. Mosses, which we remove from designated plots, and dwarf shrubs, which we remove. So we have the full combinations of these. And this is species removal. We have 3 species, and we have removals of all of these 3 species in all the possible combinations. This shows the plant biomass after 14 years, although after 20 years, the vegetation, the results are fairly similar. What this shows is for diagram's not as complex as it looks. The y-axis is standing biomass, which is the amount of carbon stored in the system. By the way, I think there's more people waiting to be admitted. Yes. So the y-axis is the amount of carbon stored above ground in the shrub layer. X-axis is the vegetation diversity, going from low diversity to high diversity as a result of our removal treatments. So this shows small islands, and there's not much difference between the treatments. If we go to medium and large islands, there's big differences between treatments. Loss of biodiversity affects above ground carbon storage in some contexts. Big islands and medium islands, because biotic factors are important, not so much on small islands. Now, we've also looked at the temporal patterns of biomass storage. And this represents an analysis that was headed up by 2 of my former postdocs, Paul Cardle and Nicola Fanine. What they looked at here was basically temporal stability of above ground carbon storage. In other words, doesn't vary much over time. If it varies less over time, it's more stable. Same story as before, stability on the y-axis, diversity on the x-axis. And it's apparent that if you remove vegetation and make it less diverse, there's not much difference among the small islands, but medium and large islands, loss of biodiversity, you end up with destabilizing effects, big differences among treatments. We've also looked at below-ground processes that lead to carbon storage. This is one such process. This is actually plant litter decomposition, which affects soil carbon storage. This is from our year 7 data, though our year 20 data shows much the same trend. What this shows is 4 treatments which have particularly big effects: root removal, shrub removal, and removal of the 2 vacciniums. Large islands, loss of biodiversity, we get big effects. Small islands, not so much effects. So just like what we observed above ground, below ground processes that affect carbon storage depend on where we lose the diversity from. Not so much on the small islands, but much bigger effects on big islands. Now, so far I've just talked about individual functions that lead to carbon storage, but in fact there's many functions. And this is something that Nicola Fanin, when he was working with us, explored in more detail through what we call multifunctionality indices. So these are indices that consider multiple functions simultaneously. Basically, this is big islands, middle-sized islands, small islands, and the diversity gradients, high diversity, low diversity. So if you lose diversity, you're losing multiple functions, not just single functions. This index goes down. But the other important point is that this curve, and this curve are actually the same. There's no effect of island size on the effect of diversity loss of multiple functions. There is on single functions, but our view is these functions all cancel each other out. And when you consider multiple functions simultaneously, there's a lot of cancelling out of different functions going on. So I've gone on a bit of a tangent here to talk about diversity. I could talk more about it, but I don't have 3 hours, so I'll move back to the main axis of my presentation and start talking more about soil aging. In other words, is what we're observing in our Swedish island system typical of other systems around the world? So what we're looking at is chronosequences, sites of different ages since they formed. And we've got 6 of these systems, 5, the Swedish one, and 5 others. And they're distributed globally, boreal, temperate, and subtropical. And they're all long-term. They range in age between 6,000 and 4 million years. And they all have relative constancy of all drivers, except the disturbance that set the chronosequence in motion in the first instance. So I'll walk you briefly through them. The first is the Swedish island system, young surfaces, productive forest. If they don't burn for a long time, don't disturb, you get much less productive. Glacier Bay in southern Alaska, surfaces of different ages caused by retreat from a large glacier, young surfaces, big trees, old surfaces, much less biomass. The Franz Josef Glacier chronosequence in southwest New Zealand, surfaces of different ages, again caused by glacial retreat, young surfaces, high biomass, old surfaces, much less biomass. The Waitutu marine terrace system in southern New Zealand, surfaces of different age caused by uplift of land from the ocean, young surfaces, high biomass. After 500,000 years, the soil struggles to maintain a forest. And 2 subtropical examples, the Kalula sand dune system in Queensland, Australia, just opposite Fraser Island, surfaces of different ages caused by aeolian movement of sand, young surfaces, high biomass, old surfaces, much less biomass. And finally, a rather well-established chronosequence across the Hawaiian island chain, across a volcanic system, young surfaces, high biomass, 4 million years since disturbance, much less biomass. So this summarizes in a very brief way the sort of initial results we got from the system. Basically, the x-axis here is time since the chronosequence started. The y-axis is tree basal area, which is simply a crude measure for some cross-sectional area of all the trees. There's a crude measure of tree biomass. So for each of these systems, Australia, Sweden, Alaska, Hawaii, and the 2 New Zealand ones, between 2,000 and 12,000 years, we have a sharp decline in biomass in each case. Contrary to what some textbooks tell us, there's no such thing as a climax community that goes on forever and ever without disturbance. eventually these systems actually degrade. And this degradation is linked to changes in nutrient availability. So what this shows, as before, same x-axis, time since the disturbance, y-axis is N/P ratio, nitrogen to phosphorus ratio of the substrate in which the plants are growing. The dashed line represents a type of Redfield ratio above which we expect phosphorus to be limiting relative to nitrogen. So for each of these systems, the points go above the dashed line at between 2,000 and 12,000 years, which is the time that the biomass decline occurs. So the biomass decline appears to be due to limitation of phosphorus relative to nitrogen. Now we've looked at 6 systems that are moderately similar, low diversity, moderately high rainfall. But there's been more analyses that have been carried out across a wider range of conditions. This is a study that was published actually last Friday that both Kenny Ning, who I guess is somewhere in the audience, and myself contributed to, looking at a much wider range of sites around the world. And we find even with wider range of sites, we still get generally, though with a few exceptions, this increase in N/P ratio, meaning that there's a rather recurrent theme of phosphorus being the limiting nutrient when it comes to long-term soil aging. Now, we also have corresponding effects below ground. We've done quite a number of studies along this line in the last 15 years. I won't give any data, I'll just summarize basically what we generally find. And that is that as the soils age and plant biomass declines, we get a reduction of the soil microbes, the bacteria and fungi that carry out decomposition. Increase in fungi relative to bacteria. Fungi are associated with nutrient-limited conditions. Declines in litter decomposition. Leaves break down more slowly. And increasing limitations of decomposers by phosphorus relative to nitrogen. Now, this last point is a particularly important point because in any chronosequence, we have a fixed amount of carbon in the rocks, and this phosphorus is finite. Eventually, it must get to limiting levels without disturbance. Nitrogen, on the other hand, can be biologically replenished by biological nitrogen fixation, bacteria that convert nitrogen in the atmosphere to plant-available forms. And contrary to what we taught in undergraduate textbooks, this phenomenon is not restricted to pioneer communities. In fact, it occurs quite abundantly also in late successional systems. For example, in our Swedish island system, the main nitrogen input is through cyanobacteria that live in a mutualism with the feather mosses. They live in a mutualism very much as clovers live in a mutualism with rhizobium bacteria. So the bacteria fix nitrogen and give it to the plant. This shows the nitrogen input through cyanobacteria are associated with the 2 dominant moss species, Pyropsium and Helicomium. And even in these late successional systems, small islands, we get quite a lot of nitrogen fixed. Whether or not it's available is a different issue, but we do get lots of nitrogen being fixed. Now, there's been quite a lot of work on these sort of chronosequences in temperate regions, but there's far less known in the lowland tropics, despite them occupying a large area of the world, and despite the fact that we think that probably lowland tropics behave differently to other systems because they've never been under glaciation, they've got ancient soils, we expect the phosphorus to be particularly limiting, but also the plants have an opportunity over evolutionary time to be adapted to low phosphorus. So several of us, myself, Ben Turner, and many others, have been on the quest to try to find a good lowland tropical long-term chronosequence. And we think we have actually found one. And this is actually the Peat Dome system in Brunei that Alex Cobb and Charlie Harvey and Sean and others have been working with for a long time, in which myself and others have been more latterly involved. These are ecosystems that have developed in back mangrove ecosystems. And over time, over thousands of years, they developed these large peat domes with really deep organic matter or humus layers in them. These can be up to 14 meters in depth. So over time, the tree roots get physically separated from the rocks which have the phosphorus in them. So over time, the tree roots and the rocks get separated, so we'd expect phosphorus limitation to occur. And this is apparent in this pollen diagram here, produced in the year. There's of particular significance of two folk in the forest ecology group. So what this shows is the succession of vegetation through peat development. So we started with peat mangrove in this pollen diagram, through to the peat formation, and then through various phasic communities. There's 2 species I want to focus on. The first is Shorea albida. In the peat domes that Alex and Charlie and others work on, they're dominated by Shorea, which is a massive tree. It goes about 50 meters in height, and it's a dominant feature of the landscape. We believe, however, that as nutrients get more limiting and they can't access phosphorus from the rocks, these large trees can no longer persist, and we end up with a replacement by a different tree, Compressa carpus, which has a very different nutrient strategy, is much smaller statured, and far more open and less productive environment. This is at the stage simply a working hypothesis, but it's one we hope to test fairly soon after the borders sort of show some permeability. Now, the other issue that's been only recently addressed with these chronosequences is whether, in fact, these effects occurred similarly across different climates. We'll just briefly introduce some work we've carried out in collaboration with Etienne Lalabute's group in Western Australia. So we have 4 chronosequences across the west coast, the southwest, and they're all long-term. They're all about 2 million years old. So we have a dry chronosequence, so dry that there's no trees anywhere near, a very wet one with really big trees, and 2 that are all intermediate. Now, these are all formed on the same parent material. This shows what we find for leaf area index, which is simply a crude measure of how much plant material there is. What we find for the wet site at Warren, then we get the classic hump relationship that we find in other moist sites, decline of biomass in the old soils. Not so much in the really dry soils, and intermediate patterns for the intermediate soils. So it would seem that moisture trumps the effects of soil infertility across this gradient, not so much across this one. And the same also with regard to soil biota. This shows soil fungi, the wet site, kind of a hump, far less so for the dry site. Similarly, meanwhile for bacteria, we find similar patterns for all the sites. So we get different things going on for bacteria and fungi. But fungi carry out most of the nutrient cycling, and it seems that they, just like the plants, tend to be more responsive to soil fertility in the wetter environment. Okay, I've now started talking about climatic gradients. So I'll keep on through this theme by now talking about temperature gradients and move into the final part of my presentation. So what we're interested in is understanding how temperature through elevation affects ecological processes. If we try to understand global warming effects, we've got 2 main empirical approaches at our disposal. The first of these is a formal experimental approach. We set experiments up to manipulate temperature, like these open-top chambers, which are commonly plugged around the Arctic to warm the ground. They're a bit like a greenhouse. They warm the ground up by about 3 degrees, or more elaborate, you can have these fancy infrared lamps and soil heating cables, or alternatively, mesocosms like this series of ecotron chambers at the CNRS in Montpellier. Now, these have informed us enormously about how warming affects physiological processes and short-term processes and mechanisms involved. But where they have a limitation is they're short-term. They represent a step change in temperature, whereas climate warming occurs generally over decadal time frames. So an alternative approach is natural environmental gradients of temperature, such as elevation. And it's relevant in this context that an elevational range of around 500 meters is comparable to the sort of change they would expect through climate warming through the remainder of the century. Now, elevational gradients have been used quite a lot in range expansion studies, population studies, but only comparatively recently in ecosystem studies. This represents 3 sites which have been used rather a lot for ecosystem studies. The sites we work with in Abisko in the Swedish Arctic, sites in Colorado, and there's also a well-established gradient along the Peruvian Andes. So the idea is you could look at processes across different elevations, and then you use the lower elevation to predict what the higher elevation will turn into as a result of global climate change. Now, our earliest work in this foray in this area is in the Abisko region in the Swedish Arctic. This is an area which is being disproportionately affected by climate change and is expected to warm by about 5 degrees in the next 80 years. What this shows is 3 of our sites, high elevation, low, medium and low, and we predict that over 80 years, this site will become more like this site with the nice purple flowers in it as a result of warming. This shows some of our very earliest work in the system, led by our then PhD student, Maya Sundqvist. And what this shows is the nitrogen to phosphorus ratio, both in the leaves and in the litter are for several plant species. 3 shrubby species, Betula, Impetum, and Vaccinium, and 2 herbs. And there's a recurrent pattern that for each of these species, we have lower N/P ratio at the lower elevation than the high elevation. This means at the high elevation, we have more phosphorus limitation than nitrogen limitation, or if we warm it up, more phosphorus than nitrogen limitation, and as we warm things up, we get less limitation by nitrogen, presumably because nitrogen gets mineralized. Now, what we're interested in is whether in fact the patterns we find here occur in other parts of the globe. So we carried out a study that was run by my former postdoc, Jordan Mayer, which involved looking at 7 different locations around the world where there are well-defined elevational gradients. British Columbia and Colorado, the Alps of Austria and Switzerland, Hokkaido in Japan, New Zealand, Kosciuszko in Australia, and southern Patagonia. For each of these locations, we set up 5 replicated transects that transcend the alpine tree line. So this design enables us to look at whether in fact the effect of warming go from here to here is simply because we have trees versus no trees, whether there's an effect of elevation independent of trees, and whether these effects are recurrent globally. So this shows what Jordan found for plant nitrogen. This is foliar nitrogen and root nitrogen. The different colors are the 7 different locations going from high elevation to low elevation. So as you go to low elevation, there's got more nitrogen in the plants, no great surprise. It's warmer, nitrogen is released more in the soil, plants take more nitrogen up. Phosphorus, on the other hand, showed some rather more sort of unpredictable trends. And as a result, when we consider the N/P ratio, we end up with what we call a phenomenon of what we call stoichiometric convergence. What this means simply is the ratios of nutrients converge at higher elevations among locations. So same as before, every color is a different region. They all have similar values at high elevation, particularly for the foliage and to some extent for the roots, and they diverge when things get warmer. Same here, if we look at the coefficient of variation among locations, they decline among locations at high elevation. This is because at high elevation, physiological processes constrain the N/P ratio. These are constrained by cold temperatures. As you warm things up, these constraints are less important, and regional factors and regional differences therefore become more important. So as a result, you find that things such as that vary among locations like parent material, geology, rainfall, the types of vegetation you have become more important at lower elevations. If you have global warming, you go from here to here, the regional factors we predict will become more important over time. We are also interested in the mechanisms that influence the N/P ratios across this gradient. And to do this, we carried out a structural equation model exercise. Now, this is simply a type of path analysis, which works out the most important possible causal pathways. So the model we tested was elevation, tree presence and absence, soil fertility, microbes, soil nutrients, and plant nutrients. This is the model that the most parsimonious model that we found across the 7 regions, and therefore is the model that we're going to go with. It shows that elevation has an important direct effect on plant nutrition, but there's also important indirect effects that are manifested through trees, organic matter, microbes, etc., all of which influence nitrogen, phosphorus, and its balance. So the investigation of these 7 regions shows that there's this decline in plant nitrogen, but not phosphorus with increasing elevation. And this leads to convergence of N/P ratios across regions. As things get colder, you warm things up, they diverge. These changes are closely linked to changes in soil nutrient availability, organic matter quality, and importantly, the nature of the soil microbial community. And also, there's quite some consistency in the responses across contrasting temperate regions worldwide. Okay, just before I finish, I'm just going to make 2 final points about these global climate studies. The first of these is that so far everything I've spoken about is really observational studies. And while they have certain strengths because we can look at long-term patterns, they also have problems with potential confounding factors. So the best of both worlds is to combine observational and experimental approaches. And this is something we're now doing through the so-called WARM network, which involves a series of sites throughout the world that we're running together with our colleagues in the University of Michigan and Sweden and elsewhere, as well as, of course, various regional collaborators. And what we're doing for each of these locations is we have a well-developed elevational transect. We have sites at high and low elevation. For both low and high, we carry out experimental warming through these open-top chambers. As I mentioned before, mini greenhouses that warm the ground up by about 2 or 3 degrees. And we also factor this with removal of dominant plant species. So we can look at the interactive effects of the vegetation and warming. And what we're interested in is how these interactions affect carbon exchange between carbon between the plant community and CO2 in the atmosphere. In other words, how carbon exchange is affected both by warming and dominant vegetation and their interaction. This is early days yet, but hopefully if I'm giving a seminar in 3 years' time, we'll have a bigger story to tell about this. And the final point I'd like to make is that these types of environmental gradients serve as a really valuable tool for undergraduate teaching. Sean and myself, together with Kang Min and Wai Kit, and of course our awesome Taiwanese friends and collaborators, run the 3rd and 4th year field course for our ASE ecology students, which we named Forests of Taiwan, Tropics of the Tree Line. A large part of the course is focused on an elevational transect through the Taroko Gorge that goes from lowland tropical forest near sea level to a tree line that consists of rhododendrons and dwarf bamboos. And you can drive through this amazing gradient in the space of 2.5 hours. So in 2.5 hours, you can transcend the same climatic range as you'd find through about 45 degrees of latitude. So we have the students measuring stag characteristics, leaf traits, soil properties, nutrient cycling, epiphytes, anything else we could think of across these locations to get some insight as to what would happen if these colder sites became warmer through climate change. This shows what the course looked like in the first couple of years. Students happily measuring vegetation and plots at mid-elevation here, and a rather cold and rainy day at tree line here. So this is what it looked like the first 2 years. For this year, for obvious reasons, the Taiwan field course took in a slightly different format, but we had a lot of fun anyway, as one does during Zoom meetings and lockdown. But nevertheless, we're very much hoping that maybe in a couple of years, the course could go back to something that looks a bit more like this. So just tying up with a few, just 3 take-home messages. Firstly, one recurrent theme has been understanding what happens in terms of linkages between above ground and below ground organisms. These are the things we see, these are the things we don't see. But understanding both components and how they interact are necessary for understanding both the biotic and abiotic drivers of ecosystems. Secondly, understanding these linkages helps our understanding of global change effects over long timescales, including much longer than that of our own experiments. And this is important because, of course, vegetation and organisms just generally are the mediators of carbon exchange between the terrestrial environment and the atmosphere. And thirdly, all the processes we look at operate over small scales, but demonstrate important feedbacks, both as drivers and responders, over, with processes that occur over much longer scales. Here's the diagram I started off with. We look at processes here, but we can generate a lot of insights by these sorts of studies in terms of what happens in vegetation change, soil processes, and geology, and such like. And on this note, I'd like to just finish by thanking the various collaborators and colleagues who have contributed to the data and ideas that I have given in this presentation. Thank you.